So I'm Elias. I'm working with the Roy Group at University of Wisconsin-Madison, and I'll be presenting the early stages of our work to benchmark different machine learning methods for predicting genomic structures within and across species using high-C data. So 3D genome organization refers to the way that DNA is systematically folded inside of a cell's nucleus. This folding allows regulatory elements to interact with their gene targets by being physically close to them in three-dimensional space, despite the fact they might be far apart from them along the DNA sequence. We can study 3D genome structure using high-C data, or high-throughput chromosomal conformation capture assays. And these represent genomic structures using contact heat maps, where the color intensity corresponds to the propensity of bins in the genome to interact with each other. So the rows and columns, the color intensity at those positions corresponds to the likelihood that these regions physically interact. high-C data is very popular, and it's growing more and more popular. Shown here, we have a data portal from the 4D nucleome, and high-C data corresponds to the tallest bar. So it's very popular. high-C data provides us with the opportunity to study structural patterns across multiple scales. So if we take one of those matrices, because they're symmetric, we can cut them in half. And then here we have 3 of those turned on their sides, and they each show a different scale of interaction. So starting on the left, the largest scale, we can see things like compartments, which correspond to regions of open or closed chromatin. And then if we zoom in along the diagonal, we can start to see things like topologically associating domains, or TADs, which are regions of DNA. Oh, sure. Thank you. Sorry about that. So topologically associating domains, which are regions of DNA that like to interact within themselves, but not apart from themselves. And then if we zoom in even further, we can start to see things like the chromatin loops, which enable interactions like those with regulatory elements with their gene targets, like I mentioned before. In order to see this level of structure, though, we need very high resolution data, and this is relatively expensive to generate. high-C data is important because it allows us to study the structure of DNA, and the structure of DNA is very closely related to biological change. These changes can do things like cause disease phenotypes, cause changes in how cells develop, or even biological evolution. And using high-C data, we have a way to analyze these structural changes. There are several relevant machine learning tasks for high-C data. Here I list 3: loop prediction, TAD prediction, and count prediction. But if you're interested, I encourage you to check out the review shown at the bottom left of this slide. But for the remainder of this talk, we'll be focusing on the count prediction problem, which is where we try to predict the matrices directly. Methods for count prediction have evolved over the years. There's a whole range of methods which can be grouped by the type of input they take in and the kind of output they produce. The earliest methods predicted counts for pairs of regions at a time. But as technology has progressed, deep learning has enabled prediction of larger structures like stripes and blocks. The earliest deep learning methods generally only took in sequence information to make the count prediction, but the most recent methods can also use things like accessibility information, histone marker binding sites, or histone markers and transcription factor binding sites, or architectural proteins. So the goal of our project is to benchmark how all these different kinds of methods perform and why they are able to perform the way they do. So yeah, we'll look at 3 main questions. How well do deep methods perform in comparison to shallow methods? Can any of these models generalize to predicting high-C in species that they were not trained on? And then which are the most important input genomic features for any model to make high-quality count predictions? So for this study, we just used endothelial cells. So it's important to remember that this is just in one biological context. But we used data from human, rat, and cow. And then we compared a bunch of different methods to high-C reg, which is our kind of like our shallow baseline. We made 2 kind of custom simple deep models, which I'll just mention briefly and then not talk about again because they're not quite performing yet. And then we compared to two more complex recent models, Epiphany, which is based on an LSTM with the GAN head, and then C Origami, which uses a transformer. We evaluate these models using two different kinds of metric. The first one is basically just raw performance, area under the distance stratified correlation curve. So it's basically a way of summing up how these models are making predictions across all different distances in a single number. And I can get more into this stuff if people are interested at the end. It just takes a little time to explain. But so this is a measure of raw performance, and then we have loop calling performance, which measures these models' ability to predict like the most important genomic structures represented in the high-C contact maps. And then for each class of model, we'll see how well they do when they're predicting on holdout sets within species, and then also on those same chromosome holdout sets across species. So we'll start off by looking at how deep methods could perform in comparison to shallow methods. So here we have the on-species prediction performance for 5 different methods. So models trained on human data, predicting on human data, rat to rat, cow to cow. In each case, it's very clear that C Origami, which is the transformer model, performs the best. But in rat data, the second most complex model is actually outperformed by the shallow method. So basically, the takeaway is that deep models generally do perform better, but that might not always be true. So there's certain biological contexts where they actually perform worse. In addition, if we look at the other way of evaluating these methods' performance, the story is quite different. So once again, we have the deep models, which in this plot are shown in blue and orange, outperforming the shallow method in almost every case. But Epiphany, which was kind of the medium performing model previously, is now performing very well in every cell type. So even though deep models are better at count prediction than shallow methods, there may be more to the story than just raw performance. Now we'll look at how these models can generalize to predicting high-C in new species. So here we have relative performance for models trained on human and then predicting on each of the 3 target species. So the leftmost plot here you've seen before. This is an on-species prediction task. And this is all measured by the raw performance score that we have. But it's very clear that when all 3 of these methods are predicting across species, they're performing much worse. And the most complex model is performing relatively very much worse, although a lot of that is due to the fact that it's performing so well to start with. So here we zoom out to all training and testing pairs. So the previous slide is now the top row, and on-species prediction is shown down the diagonal. So we're interested in looking at the plots on the off-diagonal elements. And we can see that in almost every case, all of the methods are generally performing much worse, with the most complex model doing the best. But in models that are trained on cow, that is actually no longer true. When we do the same thing but measure performance by loop prediction, we can see that the deep models are actually able to predict loops even across species. And once again, Epiphany stands out as the best at this task in almost every case, which reiterates that there's something about this particular architecture that makes it very good at predicting the most important genomic structures. And our last question is, which input features are the most important for all these models to predict high-quality high-C counts? So what we did was we trained each model with different combinations of the input features. So we're basically doing a feature ablation study. And we used accessibility information, architectural protein binding sites, and histone markers, and then different combinations of those. And we're comparing the model performance to models that were trained with access to all the different types of information. So here we have on-species performance for C Origami, the transformer, and Epiphany, the LSTM/GAN. And we can see that, especially in C Origami, the bars that correspond to models that were trained without access to architectural proteins, so the blue, green, and purple bars, are having the greatest drop in performance relative to the pink bar. And that's true across all species, but a little bit less so in rat. So something about rat makes it a little bit more robust to changes in input information. And the same kind of pattern is held out for the LSTM model. Also, models that are trained with only access to accessibility information generally perform much worse across the board. When we look at loop detection performance, we see a similar pattern where the models seem to be a little bit dependent on the presence of the architectural proteins. But there are also some other interesting situations that come up. For example, Epiphany, the LSTM, seems to be able to predict these most important structures almost regardless of the kind of information that it's given. So this architecture is very, very good at predicting like the most important structures in the high-C contact maps. In addition, there's some like other interesting little features that pop out, like, Epiphany does even better than anything else when it's only given accessibility information in rat, but in human, C-origami with that same input feature set doesn't do anything at all. So there's definitely some like interesting patterns popping out. And then when we compare off-species prediction for all the different models side by side, there's no clear pattern. So I'll come back to that. But in general, it seems that the models perform better when they have access to more information, which I guess is somewhat obvious. But I think the message is that how the models do and which features they depend on are very dependent on the specific biological context as well as the specific model architecture. So in conclusion, the best performing methods often depend upon the choice of metric for how you evaluate them, as well as specific biological context, be that species or possibly cell type. And then using architectural proteins can be very important for models to make good predictions on species, and that all types of models are very bad at generalizing across new species. I'd like to say thank you to the Roy Group for all of their support, and Armando Serrato, who did a lot of work for this project. Thank you.
Question for Elias. You can come here or you can say the question and you can repeat the question.
Yeah, I think we're going to set it up so you don't need to repeat the questions.
No, that's going to have that lunch, sorry.
So unfortunate thing about genomics is if I do an experiment on my sample and you do an experiment on a related sample, they're just going to look different. There'll be some form of batch effect or subtle difference. Have you looked at how much that can affect things? Because one major difference between the species is just that it's a different sample and a different experiment. And I'd be curious how much of the degradation of performance is related to just not having like the exact same experiment characteristics.
So the question is how much of the model performance is dependent on specific experimental features? And I actually want to clarify a little bit. Do you have an idea of how you would do that?
If you had just replicate experiments done by different groups, ideally.
Okay. Yeah, so we have not done that, but I think that's a really good idea. So we definitely want to look at kind of stuff like that, doing averaging across different kinds of inputs and especially across cell types. Also multi-species training. There's a lot of different combinations that we want to explore still. So that, yeah, this is early stage. Thank you.
So can you determine what is actually going on when the shallow data sets outperform the deep ones, and what patterns might exist beyond, say, being in cows in your example? What could we get maybe to augment the deep models from the shallow models?
To some extent we can. We can look at the... Oh yeah, sorry. So the question is, can we look at why the shallow methods might be outperforming the deep methods in some cases? So we can look at particular situations for the shallow model, because it's interpretable by nature. We can look at like the weights within it. The question is why would the deep model be performing so much worse? And that's a very interesting question. And we definitely want to get more into that so we can do some things like saliency mapping, like looking at which specific input features. A lot of the stuff that the keynote was talking about can come into play there, but we have not done that yet. Thank you.
We have time for one more question. I'll ask you my question.
Sure.
What happens when you combine the models? Do you, like basically if they predict differently on different types of features, maybe combining them would give a boost in performance?
So we haven't tried that. So the question is, how does the performance change if we combine the different models? We would have to think of a good way to actually combine them. I don't know how I would do that just like right off the bat. It's very specific. So one thing I didn't mention is that these models all have a different kind of output. So they're predicted as very, very specific size. The deep models in particular are very different from how the shallow method predicts. So actually combining them would be like a non-trivial thing. But I think it's a good idea to like actually combine the models in addition to the input features.